Good morning to our lecturers and my fellow friends. My name is Kaniyandan Karthikesu and today we are going to discuss about Taenia Saginata. What is Taenia Saginata? Taenia Saginata is commonly known as beef tapeworm, is a zoonotic tapeworm. It is an intestinal parasite in humans causing taniasis, a type of helminthiasis. Epidemiology of this tapeworm. These tapeworms that can cause taniasis are found worldwide. For example, Taenia saginata, Taenia solium, and Taenia asiatica. Eating raw or undercooked beef or pork is the primary risk factor for acquiring this taniasis. Infections with Taenia saginata occur wherever contaminated raw beef is eaten, particularly in Eastern Europe, Russia, Eastern Africa, and Latin America. Taniasis due to Taenia saginata is rare, except in places where cattle and people are concentrated and sanitation is poor, such as feedlots of cattle can be exposed to human feces. Human Taenia saginata infections are restricted to definitive adult tapeworm stage, which has more limited public health burden compared to Taenia solium taniasis. So in this graph or this map, we can see that so we can see that the reports of Taeniasis due to Taenia saginata and Taenia species in the period of 1990 to 2017. So we can see here that the red color spots are due to the Taenia saginata Taeniasis. So this red color, it's almost nearly the European countries and also the Latin America countries, or including Russia also. Mode of transmission. How this Taenia saginata transmits. The infectious agent of Taeniasis is Taenia saginata. Mode of transmission is by cattle to human via ingesting raw or improperly cooked beef. There is no person to person transmission in this Taeniasis. How this Taenia saginata enters the flesh of these cattle we will discuss this in this life cycle of it. Humans can are infected by ingestion of raw or undercooked beef infected with Cysticerus bovis, the larval stage of Taenia saginata. The incubation period is variable. Eggs occur in stool 10 to 14 weeks post-infection. About this post-infection, we will also discuss in the life cycle. Communicability. This Taenia saginata is not directly transmittable from person to person. Eggs of Taenia saginata passed in the fishes from men are only infectious to cattle. Adult tapeworms may persist in intestines for up to 30 years and are able to disseminate eggs for all of this time. Eggs may remain viable in the environment for months or even weeks also. The cycle of this infection repeats when infectious eggs are passed into the fishes and later ingested by the cattle, slowly migrating into the flesh and transforming into the larval stage. So this is the life cycle. So about this life cycle, I will be using the diagram at the third slide of this. So we can have a glance through of this. Okay. So here, the stage starts from here. Taniasis is the infection of humans with the adult tapeworm of Taenia saginata or Taenia solium. Humans are only definitive hosts for Taenia saginata. Eggs or gravid prokaryotes are passed with fishes. The eggs can survive for days to months in the environment. Cattle and pigs become infected by ingesting vegetation. Here, this part are infected by ingesting vegetation contaminated with eggs or gravid prokaryotes. In this animal's intestine, the oncospores hatch and invade the intestinal walls and migrate to the striated muscles where they develop into cysticerci. A cysticerus can survive for several years in the animal. Humans become infected by ingesting raw or undercooked infected meat. In the human intestine here, the cysticerus develops over 2 months to an adult tapeworm, which can survive for years. The adult tapeworm attach to the small intestine by their scolex. Here shown in the diagram, we can see the scolex and reside in the small intestine. Length of the adult tapeworm is usually 5 meters or less for Taenia saginata. However, it may reach up to 25 meter, and the adults produce prokaryotes which mature and become gravid, detach from the tapeworm and migrate to the anus or are passed in the stool, approximately 6 per day. The Taenia saginata adults usually have 1000 to 2000 prokaryotes. The eggs contained in the gravid prokaryotes are released after the prokaryotes are passed with the feces. They are passed in the feces. And Taenia saginata may produce up to 100,000 eggs per prokaryote respectively. So that is the life cycle of this Taenia saginata. So let's go to the clinical features of teniasis. Teniasis is an intestinal infection, as we all know, where Taenia saginata teniasis is usually asymptomatic. Mild and non-specific symptoms are usually visible in highly infected individuals. Symptoms develop about 8 weeks after ingestion of meat containing cysticerci. Abdominal pain, nausea, diarrhea, or constipation. Prokaryotes appear in the stool and protrude from the anus. As I told in the life cycle, we saw that the prokaryotes appear in the stool. Infected people may develop visible or palpable nodules beneath the skin. Symptoms may continue until the tapeworm dies following treatment. Otherwise, it may live for a number of years, up to 30 years actually. So the laboratory diagnosis of this Taenia saginata is we can use morphological species differentiation is often not possible based on the gravid prokaryotes, but can be done by DNA analysis, PCR, polymerase chain reaction. Naked eye examination of stool or for prokaryotes. It is easy to diagnose if the 1.5 to 2 cm long of 0.7 cm wide segments are eliminated in stool. Taenia saginata eggs are shed irregularly in stool and cannot be differentiated morphologically from Taenia solium eggs. The third method we can use is direct smear microscopy for eggs. The fourth method is cysticercosis can be diagnosed by biopsy examination of nodules, X-ray, MRI or antigen by ELISA. Using an ELISA Coproantigens are detectable in stool fluid even when neither proglutids nor eggs are being excreted. Another diagnosis we can use is eosinophilia. An elevated IgE levels are chief hematological findings. Also, zeal and Nielsen stain can be used to differentiate between mature Taenia saginata and Taenia solium in most cases. Taenia saginata will stain while Taenia solium will not, but the method is not strictly reliable. So these are our references. So if you have any doubts or if you have any queries, you can just go to the website and also search for more information if you like to. And thank you very much for listening, and thank you.